In the flow of genetic information, we say that DNA goes to RNA, which goes to protein. So we want to look at now this RNA component. And as I mentioned in the previous video, in terms of the flow of genetic information, the messenger RNA, which is the specific one kind of shown down here, is really the one that leads to the formation of protein. But in more general terms, I want to talk a little bit about RNA, or ribonucleic acid. So there are differences between DNA and RNA. So the one we talked about in the earlier part of this chapter is that in RNA, the sugar is ribose instead of deoxyribose, right? So it would have an OH group on that second carbon of the sugar. And the other thing we talked about already earlier was that in RNA, you're going to have uracil instead of thymine as one of the bases. So in RNA, you're going to have A, C, Gs, and Us found. So if you see a nucleic acid structure that has a U in it, it means you're looking at RNA. If you see a nucleic acid that has a T in it, you'll have DNA. And you can't have one that has both U's and T's. RNA is going to be a single-stranded molecule, so while DNA is a double-stranded molecule, like we looked at with the double helix, RNA is just going to be a single-stranded molecule of nucleic acids. And it's also going to be smaller than DNA usually. So chromosomal DNA, as we mentioned, was in the millions of bases or nucleotides long, whereas RNA is usually maybe like in the thousands or so. All right, so there's three main types of RNA molecules. You have ribosomal RNA, or rRNA. That ribosomal RNA is important for making your ribosomes, which is where protein synthesis occurs. Messenger RNA is the molecule we talked about that serves as that link from DNA to protein. Messenger RNA is that link in the middle. And then transfer RNAs are these important molecules that help build a new protein chain. So the transfer RNAs, they transfer in new amino acids into a growing protein chain. And we'll look more at those whenever we talk about translation. Okay, so I mentioned most of these things on the previous slide. The tRNA is typically drawn as this cloverleaf shape, so it almost looks like a clover, and there's a picture of it here where you can kind of see the different features of it. Now, the most important part of a tRNA is probably this anticodon down here. This is the part that is going to bind to your messenger RNA and basically helps determine if the anticodon binds to the messenger RNA, then a particular amino acid is going to add. And we're going to look at how this works in a little bit more detail coming up here in just a minute. All right, so again, the types of RNA, the rRNA, the messenger RNA, and then the tRNAs, they all have their own unique functions, and the tRNA is going to be the one that specifically binds to the messenger RNA and brings in a new amino acid. Okay, so let's look at how these molecules work together to undergo transcription. So transcription is going to be the synthesis of messenger RNA from DNA. So this is that process where we go from DNA to messenger RNA. That process is called transcription. Okay, so as we talked about, that the DNA is a double helical structure. So in order for this synthesis of the messenger RNA to occur, DNA has to split and separate into two strands. So it kind of has to unwind. Once it unwinds, one of the strands, and it's specifically the strand that goes from 3' to the 5' end. So I'm going to draw up here. If you have a DNA that goes 5' to 3', and then the other strand goes 3' to 5'. Whenever those two separate, this one down here is going to be the one that is used for transcription, right? So it's the one that goes in that 3' to 5' direction. And then what happens is there's an enzyme that's going to come in, and it's called RNA polymerase. So RNA polymerase is going to bind to this template strand. It's going to bind here, and it's going to basically walk along the molecule this way, and it's going to make a messenger RNA molecule that I will draw here in blue that is going to go from a 5' to 3' direction of a new messenger RNA. So your DNA is the double-stranded molecules that are in red up here, right? This double-stranded molecule. This one that goes in the 3' to 5' direction here is going to be your template strand from which the messenger RNA, which is in blue, is made from. So if you look down here at this bullet point, it says transcription forms a messenger RNA with a complementary sequence to the template. So basically, if this is the template, what we mean by a complement is that if there's an A there, you would get a U in your messenger RNA, right? You usually think A goes with T, but remember in messenger RNA, we don't have T's, we have U's. So if you had, let me just write in a couple letters here. So if you had A, C, G, T on your DNA, then on your messenger RNA, you're going to have the complement of A, which would be U, the complement of C, which would be G, the complement of G, which would be C, and the complement of T, which would be A. All right? So again, you're making a complement from your template molecule. All right. Okay, so now let's look at an example here. So it says, from the template strand of DNA below, write out the messenger RNA and informational strand of DNA sequences. So the template strand is the one that goes from 3' to 5'. The informational strand of DNA, info strand, is going to be the complement strand of DNA that goes in a 5' to 3' direction. So this is going to be a DNA molecule. So that means we're going to have A, C, G's, and T's. So wherever we have a C, we have a G. A T is an A goes to a T, C, G, T, A, T, G. So the informational strand of DNA would be GAT, CGT, ATG, right there. So remember, our DNA is going to be this double-stranded molecule here. Now, for our messenger RNA, right, if I said what is the sequence of the messenger RNA, we're always going to write it 5' to 3', and it's going to be a complement of that template strand. So it's going to be almost the exact same thing as the informational strand, with the exception of we're not going to have T's. So the complement of this C is still going to be G. The complement of T is going to be A. The complement of A in messenger RNA is going to be U. And then C, G, U, A, U, G. So the one thing that you can notice from all of this is that your messenger RNA strand and your informational strand are almost identical. The only differences are wherever you have a T in the informational strand, you're going to have a U in the messenger RNA. So wherever we have a T, we have a U. Where we have a T, we have a U. Other than that, the informational strand and the messenger RNA are the same. Now, the one thing to keep in mind, though, is that whenever we make this messenger RNA, we're making it from this template strand, and we're making it by adding complementary bases, C to G, T to A to U, G to C, and so on and so forth.